evening, students. So we have completed the male as well as female reproductive system, oogenesis as well as spermatogenesis, and then we have switched to the menstrual cycle. All the 4 phases of the menstrual cycle we have completed. Now today's topic is fertilization. So what is fertilization? Basically, in simple terms, fertilization means fusion of male as well as female gamete to form a diploid cell, and that diploid cell is called as zygote. So what is fertilization? It is nothing but fusion of male and female gametes to form a diploid zygote. Diploid zygote means the zygote will be 2 and the ploidy will be double. That is, in case of humans, if we are talking about, the total number of chromosomes would be 46 or 23 pairs. Right? The location of fertilization would be the ampullary isthmic junction of the fallopian tube. That is what we have discussed when we were discussing female reproductive system. So location is very important. It is ampullary isthmic junction of fallopian tube. Now, on which day the fertilization takes place? Right? So we know exactly on the 14th day egg is released. Right? On the 14th day, ovulation phase or ovulatory phase, egg is released from either of the ovary. Right? Egg remains in the fallopian tubes for 48 hours. That is 2 days. So egg remains in fallopian tube for 2 days. That is, you can say 48 hours. So that is, on the 14th day, if the egg is released in the fallopian tubes, the fimbriae collect the egg, ovary is producing the egg. Through the help of fimbriae, it is collected and it is stored where? In the fallopian tube. Egg remains in the fallopian tube for 48 hours, that is 2 days. So if we are saying the exact location would be, or the day would be 2 days, that's it. So if the 14th day is the day of the ovulation, then either on the 15th or 16th day of the menstrual cycle, fertilization must take place if the sperm is available in the female reproductive tract. Right? Now, few facts that we have discussed before also. In one ejaculate, we know in one ejaculation approximately 3 to 3.5 ml of semen is released, and in this 3 to 3.5 ml of semen, the total number of approximate number of sperms are around 200 to 300 million sperms. So in each ejaculation, approximately 3 to 3.5 ml semen is released out, or it is ejaculated out. Right? Within this 3 to 3.5 ml of semen, approximately 200 to 300 million sperms are present. Now the next thing is, the sperm has to start its journey from vagina, that is the entry point of the female reproductive tract, and sperm has to reach to the ampullary isthmic junction, because this is the area, the location, the junction where the fertilization will take place. So now sperm has to start its movement. Now the basic thing is, when the sperm starts its movement, there are some things which work with the sperm. That means certain structures, certain chemicals will proliferate, or certain chemicals will give a positive response so that sperm reaches to the ampullary isthmic junction. But there are certain structures which act as a barrier. So let us see the structures which help the sperm and which act as a barrier. So first step is approach of the sperm, right? So first step is approach of sperm, right? This is the first step of fertilization. So two things, that is the first A, which help the sperm to reach up to the isthmic ampullary junction in order to initiate or in order to facilitate fertilization. So which help the sperm, it includes, this is first step, approach of sperm. Within first step we have A and B also. Within A, the first is the lashing movement of sperm. The lashing movement of sperm, like this, with the help of its tail. We have already discussed within the structure of sperm that they have double kern, that is the mitochondria. That is nothing but mitochondria. This mitochondria is going to provide the energy in the form of ATP, and this energy is required or it is used for by sperm in order to start its journey from vagina till the ampullary isthmic junction of fallopian tubes. The second important chemical is prostaglandins, and we have also discussed the role of prostaglandins, that is it initiates or it causes the muscular contraction, that is the contraction of the uterine wall as well as vagina. So it causes contraction of vaginal wall as well as uterine wall. Because of this contraction, the movement of the sperm starts within the female reproductive tract. The third is aspiratory movement of uterus. So it is aspiratory movement of uterus. Now what is this aspiratory movement of uterus? Aspiratory movement is upward movement, that is a kind of suction movement of the sperm. So this is upward or suction movement of sperm within the female reproductive tract. So the first part is the structures which are helping the sperm in order to reach the ampullary isthmic junction. Now the second part, that means the structure, that is A, now it is B. B, that is the structures which act as a barrier. So barrier which prevent the sperm to reach to the ampullary isthmic junction. The first important structure that is present within the female reproductive tract is the endometrium layer, which is highly thick, folded and glandular. So thick endometrium, thick and highly folded endometrium. So let us say if this is the region of the ampullary isthmic junction, and here it opens into the uterus, right? Within the uterus, the epimetrium, myometrium as well as endometrium is present. Now endometrium is highly thick, like this. It is highly glandular also, right? And it is highly folded. So this, within this tube of folded region, sperm may be misguided. So majority of the sperms get killed within this layer only. The second important, see B1. Second is acidic pH of the vagina. So acidic pH of vagina. Sperms do not act, they cannot remain live in the acidic pH. So acidic pH of the vagina kills the majority of the sperms, right? And the third is the phagocytic nature, that means the phagocytic cells of vagina. Third is phagocytic cells of vagina. What these phagocytic cells will do? Phagocytic cells will perform the function of phagocytosis. And what is phagocytosis? It is basically engulfing the sperms by endocytosis, and through exocytosis the sperms, that is the digested sperms, will be released out from the female reproductive tract. So first step is approach of the sperm. Within the approach of the sperm, what we have discussed, certain structures which promote this movement of sperm to the ampullary stomach junction, certain structures act as a barrier, right? Now with the help of this helping nature structures as well as barriers, approximately we can say 100 to 150 sperm reaches to the fallopian tubes. Right? Now 100 to 150 sperms reaches to fallopian tube, right? And sperm move at a rate of 1 to 4 mm per minute, right? So rate, that is the speed of sperm movement within female reproductive tract is 1 to 4 millimeter area, that is done per minute. This is again very important but not given in your textbook, right? So rate of this sperm is 1 to 4 mm per minute. Phagocytic cells, acidic pH, and the endometrium thickening, that is highly folded glandular cells or glandular layer of endometrium. Now 100 to 150 sperm reaches to the fallopian tubes. Now there has to be certain changes that takes place within the sperm itself. When sperm has entered the female reproductive tract, that is in the vagina, and that process, that is in order to attain the physiological maturation, the process is termed as capacitation. So we are discussing first step only, right? So first step includes, that is capacitation. What is capacitation? The process in which sperm attain the functional maturity. What is capacitation? Capacitation is a process in which sperm attains functional maturation. Right? Sperms attain the functional maturation, and these changes are the changes that takes place in the sperm cell when the sperm cell is within the female reproductive tract. So the first change that takes place is from the acrosome part, the sterols as well as glycolipid content starts deteriorating. That means, so changes in the sperm cell. The first change, that is removal of sterols and glycolipid content from the acrosome part of the sperm. Acrosome is the cap-like structure which is present in the head part of the sperm. So first is removal of sterols and glycolipids or the glycoproteins from acrosome. Now why sterols and glycoproteins must be removed from the acrosome? See, the answer is to this question is very simple. Say for example, this is the region of the acrosome, and within the acrosomal membrane these sterols as well as glycoproteins and glycolipids are located, right? In order to remove the thickness of this acrosomal layer must become weak now. Why acrosome layer must remain weak? Because now acrosome function is to release sperm license. Sperm license contains various enzymes, and these enzymes will digest the overall layer of the egg, that is zona pellucida, corona radiata, like that, right? So now acrosome structure will become weak. How it will become weak? By removal of sterols as well as glycoproteins. And why it should remain weak? Because if the layer is not disrupted, how sperm license will be released from the acrosome? So that's why this change is required. Then calcium ions also taken in, and these calcium ions increases the activity of sperm cells. So So calcium ion taken in, which increases activity of sperm cells. Right? So this is the first step, which includes the approach of the sperm. Positive approach, that is the structure which helps the sperm to reach to the fallopian tube, that is ampullary stomach junction, in order to facilitate fertilization, and certain structures which act as a barrier. Capacitation, that is the process in which sperm attain the function maturation or the physiological maturity, and these are the two changes that takes place within the sperm structure when the sperm is in the female reproductive tract. Now the second part is, that is the step number 2. Step number 2 is fertilizing anti-fertilizing reaction. See, all these things are not at all given in NCERT, so please, you have to rely on the lectures that I am providing, and do take the advantage of all the lectures. So this is step number 1, that is approach of sperm. Now step number 2 is fertilizing anti-fertilizing reaction. Fertilizing anti-fertilizing reaction. Now what is this fertilizing? Fertilizing is a chemical that is secreted by the ovum or egg, and these chemicals as spermophilic sites. That means the site where sperm can come and attach. So fertilizing is chemical secreted by egg or ovum cell. This has spermophilic sites. Spermophilic sites, and spermophilic sites are the sites where sperm can come and bind. So it is a site for attachment of sperms. So let us say if this is the membrane of the ovum or the egg, right? Then the structures, the spermophilic sites, which are the attachment of the sperms, how it is arranged? Like this. The sites are present for the sperms to come and bind. Now what happens? 100 to 150 sperm reaches to the fallopian tube. Out of 100 to 150 sperm, we know only one sperm has the capacity to fertilize the egg. So out of 100 to 150 sperm, now only few sperms can come and attach on the spermophilic sites. See, this is a natural mechanism to decrease the number of sperms, right? Natural mechanism to decrease the number of sperms fertilizing the egg or the ovum cell, so that only one sperm fertilizes the egg and only one diploid zygote will form. There should not be any polyspermic condition, so that many sperms can come and attach on the ovum. So spermophilic sites, attachment of the sperms. So out of 100 to 150 sperm cells, only 10 to 15 sperm now remains. Where? In the fallopian tubes. So this is about the fertilizing. Anti-fertilizing means what? It is a chemical which is secreted by, sperms itself. Like it is a kind of antigen-antibody interaction. So say for example, if fertilizing is acting as an antigen, then anti-fertilizing is acting as an antibody, right? So antigen-antibody interaction analogy you can understand, right? So this is step number 2, fertilizing anti-fertilizing reaction. Now what happens? Sperm has binded to the spermophilic sites, and these sites are present in the ovum, because this is a chemical that is released by the ovum itself. Now what is going to happen next? The release of sperm license from acrosome part.